Good afternoon, colleagues. Sorry I have to interrupt the conversations, but I hope that Prof. Robin Crewe's presentation will add to this conversation. And we would like to welcome all of you. There are quite a number of you that are coming to STIAS, and I hope that the few of you are coming to STIAS for the first time, and that at STIAS, when you come for the first time, you have to come back again. For those that are coming for the first time, my name is Edward Chilumira. I'm the director STIAS, and it's my pleasure to start off the conversation this afternoon. This is the first of 3 public lectures that STIAS offers as part of its fellowship cohort that is in residence, and the idea is that through these public lectures we can show the public what we're doing here, and maybe even answer to some extent, what are we good for? Robin is going to be the first to prove that we still have to exist. Just no pressure at all. But as you know that during the week we have internal seminars with the fellows on Tuesdays and Thursdays, to which some of you are invited and you can join us. But the 3 public lectures then are public to the public and are also live streamed. So in case you want to remember a certain phrase that Robin said, you can go back to our YouTube platform and replay what has been said and discussed and the questions that have been forwarded. So thank you very much, Robin, for setting the stage for 2026. And also we would like to thank Professor Teresa Vossler for accepting to introduce today's speaker, and she will welcome him for us, and then we proceed. But before I invite Teresa, I would like also to mention that it's not all talk at STIAS. After the public lecture, there will be a glass of wine outside there, so we can look forward to that. But also that there will be, as I said, 2 more public lectures that will come up. On Thursday, 16th April, there will be a public lecture by Jean-Louis Fenji dealing with sovereign AI and epistemic justice. And then on Thursday, 21st May, we will have Roseanne Kennedy from Australian National University, who is working on remembering deep pasts for the present. And so we look forward to those 2. As I said, we do also have a lot of people online, and we do also have quite a number of former fellows that are listening in, and so we would like to thank all of you. And Christoph, our senior program manager, will handle the question and answer, and will recognize some of those that are online. So briefly, if I may, about Teresa, who was hiding in the middle here. We're waiting for her outside because we normally escort our introducers. So now I think next time we're just going to sit here and let them come in. So she is in the Department of Botany and Zoology at Stellenbosch University since 2002, and has just served 6 years as the executive head of the department, and you can see that she survived. She obtained her degrees from the University of Wits and a postdoctoral fellowship at the University of Pretoria, and I think that's where they got in touch with Robin, and Robin has been her mentor for quite some time and guidance. So it's her turn now to introduce him. And so you can edit out the good things he did and mainly point out the ones that you are not very pleased with. Her research focuses on the behavioral and chemical ecology of social insects, particularly honeybees, with a long-standing interest in understanding the unique biology of the Cape honeybee. She's made significant contribution to advancing knowledge in insect social behavior and evasive biology. So I think between the two of them, they will tell us why the bees behave the way they do. And she's also led major research projects and currently serves as editor-in-chief of the journal African Zoology. So those that have not been accepted when they submitted their articles, you can look at Teresa there. So she has previously received Stellenbosch University's Rector's Award for Teaching Excellency. And so, Teresa, if you may, please.
Thank you very much. I might have survived the head of departmentship, but it didn't, my hair didn't survive the head of departmentship. So I've known Robin for a very long time. I actually know him since, no, I'm not going to give it because it'll give our ages away. But I'm going to say that somewhere in the 80s. So I've done all of my degrees with Professor Crewe. So I've done my honours, my PhD at WITS under Robin's guidance. And then I've joined him at Pretoria University and did a postdoctoral fellowship for about 5 or 6 years before moving over to Stellenbosch University. Robin is a highly distinguished scientist and is known for his chemical ecology and his behavioral ecology, especially for social insects. But other than his scientific strengths, Robin has a very strong influence and has made many changes in the higher education space. And I think that is as important as the scientific contributions that he has brought into the scientific realm. So I thought I would just start by saying, and this is, Robin, I think you have, as a mentor, I would like to actually thank you and say that I'm grateful for the way that you have guided and mentored me. You've always had that, given me space, the independence, to actually follow and grow and develop the way that we wanted or your students wanted. But at the same time, you gave us the resilience to actually, if we chose an academic pathway, to basically how we were going to make our way through that academic career, because it's really not that simple. And that resilience and that space and that independence has actually been totally, if I didn't have that, it would have been a nightmare. So Robin started his career as a biochemist. Well, that's what he did his undergrad and his master's in at University of KwaZulu-Natal. He then went across to the USA, which I thought I might add a few words of wisdom about, but then I decided, no, I'm going to put my foot in my mouth. So I really won't mention anything about the USA. But he went to the University of Georgia, where he got a PhD in entomology. And from there, he became very distinguished researcher in the field of chemical ecology, as well as behavior in social insects, mainly honeybees, lots of ant work. And this was the way, and he spent and focused on trying to understand reproductive regulation, division of labor, chemical communication. And this really has given him very strong international networks. And so he has many collaborations with national and international scientists. And by doing that, he has really added great value to the body of literature that we have concerning chemical communication. Having said this, he has been very successful in an academic career in that he has published over 165 reviewed articles. He's also, what I think is very important, so Robin has also, he is author of many popular articles. And I think it's very important for us as academics to make that, what we know, available to the public. And Robin has been doing that continually. And I think that's something that we should all strive to do. He also has many conference proceedings. And then I thought I would say how many contributions he has made to seminars, symposiums, conferences. And then I started counting, and then I gave up. Because I promise you, you could have turned old counting these lists of conference contributions. So he has really shared his knowledge far and wide. Back to what I think is as important, is his mentorship. He has mentored 35 postgraduate students. He has hosted 10 postdocs. And many of the students that have come through Robin's guidance have successfully got jobs, and they are successful in those positions. So they have followed academic pathways, many of them, but many of them have stayed in the science environment, making a difference. So I think that is something that I think is something to be proud of. But the other thing that Robin managed to do simultaneously is, besides that research and scientific endeavor, he actually had a lot of time that he put aside to leadership in higher education, which is not easy. So when he started finding an interest in leadership and governance, Robin first then went to the Business School of London to make sure he had a skill set, and then he started first with a head of department at Wits University, not 1 term, 2 terms. Then he became deputy dean at Wits. Then that wasn't enough to put him off. He took 2 terms then he went to the deanship of Wits, and during that he moved over to Pretoria University, became dean there of science and agriculture. From there, he became the vice principal for research and postgraduates. And in that capacity, he really started changing research structures, internationalization, and there's been really, Pretoria really grew from Robin's inputs during that time in those 10 years. So that he did for 10 years. And I take my hat off to Robin for managing to stay in that space for so long, and at the same time making a huge difference. And he has been recognized for that. He has been invited to many academies. So he is a fellow of the Royal Society of South Africa. He actually founded and is and was president of the Academy of Science of South Africa. He, now I've got to think, all these acronyms. Has anybody seen so many acronyms? He is a fellows of the, World Academy of Sciences, the African Academy of Sciences, and to really show the appreciation for what Robin has done for the science community. He has been really a appreciated by being awarded a number of very prestigious awards. So he has received gold medals from the Zoological Society of South Africa, the gold medals from the Academy of Science South Africa. He was the first person in Pretoria University to get the Harry Oppenheimer Fellowship Award. So that was the first person at Pretoria University. He then got the Lifelong Achievement Award from the National Science and Technology Forum. Take this. The National Science and Technology Forum is known as in South Africa, as the Science Oscars. So that's quite a big thing, eh? Science Oscars. Robin got the award there. And very recently, and this is really a very prestigious award, he received the John Herschel Medal from the Royal Society of South Africa. And that is all to honor Robin for his influence in advancing both science and the higher education space within South Africa. I can say Robin is a lifelong learner. What does he here do? He's here writing 2 more books. So Robin is not going to hang up his gloves very soon, and I am very pleased to introduce Robin and to welcome him, and we look forward to listening to your talk and to thank him for everything he's done for science in South Africa. Thank you.
Thanks very much. Thank you. Well, ladies and gentlemen, I don't know if I can proceed after all of that. But thank you very much, Teresa. It's very heartwarming to have an introduction of that kind. And I'm delighted that you agreed to do it. So thank you very much for that. It really is a great honor for me. And it's also a great honor for me, and I need to thank STIAS for that, that they took the risk. And of course, after what you've heard, this is a high risk, that I would actually be able to come and do some work on a book on the Cape honeybee. So I'm very grateful for that opportunity, and I'm also very grateful for this opportunity to be able to present this lecture today, because I think what I'm going to try to do is to give you a brief insight into how we're going to characterize the biography of the Cape honeybee. And I'm going to intertwine aspects of the natural history of honeybees and the individuals who've actually made the discoveries. And hopefully that will give it a kind of immediacy that you don't get from just a bland statement of what was discovered. So let's hope well, you can tell me at the end whether we've succeeded or not, and how we need to go forwards. In addition to that, I would like to thank a number of people who have come from some significant distances to actually listen to the talk this afternoon. My beekeeping friend John Moody, who's come from Heidelberg, and our friends from Napier, Stan and Leslie Kahn. It really is very heartwarming to have you here, and hopefully it was worth the effort. So, let me start then with the information that I want to give you. As you can see from the slide that's on the screen, I'm showing a picture of a wild honeybee colony. And that picture was taken recently about 17 kilometers from Hermanus. And it shows an extremely large colony in a crevice Some of the largest combs are almost a meter in length. So it's a huge colony. And if you think about it, it's colonies like that that actually provide beekeepers with the bees that they then use to establish their businesses. And in some occasions, they actually put those colonies into beehives. And I brought a classic beehive here just to show you that those are the kinds of boxes that beekeepers used to put bees into, but they use more efficient methods these days. The other thing is it's important to recognize that these are the colonies that also sustained the Khoi pastoralists who were here before the colonists arrived in the Western Cape. And so these are very important insects from a variety of reasons, particularly from an agricultural point of view, because a large part of the Western Cape economy, agricultural economy, is dependent on these organisms. Now, interestingly, the IUCN, which keeps the red list of threatened species, has recently added wild honeybees in Europe to that list. I don't think that's the case necessarily locally, but it does give you some idea about the importance of honeybees. All right. The other thing that I thought I would do is to just give you this quotation from William Morton Wheeler's book, Social Life Amongst the Insects, one of the most seminal books about social insects. And you can see from that quotation that he indicates that when people talk about bees, they usually mean the honeybee. And so I thought what I would do is to just give you a brief idea of what the situation with bees is, and to give you bees in context, as it were. So the first thing to recognize is that there are roughly 20,000 species of bees globally. Locally here in southern Africa, we have about 2,000 species of bees, or 10% of the total world population. Now, the number of species of honeybees is actually 12. So it's a minuscule fraction of bees in general. So there's something special about honeybees, other than that we like them and have interacted with them. And then the honeybee, which we all recognize as Apis mellifera, is a species of honeybee that is subdivided into about 23 different, well, it varies with what opinions you get, about 23 different subfamilies. But it's one species. Okay. So, let me just take, so that's the question about the honeybees. Now I want to talk about this most intimate association that bees have had with our species. And if we look in the archaeological record, one thing we find is that bees have been exploited in the Near East, in Europe and North Africa for at least the last 9,000 years. Interestingly, bee exploitation in southern Africa is much older. There's evidence that goes back at least 40,000 years. So this interaction between humans and bees has been going on for a long time. And then if you look at the picture on the left-hand side of this slide, you see a rock painting from a cave in Spain. And it shows an individual climbing a ladder and actually harvesting some honey from a bee colony in a rock crevice, and you see some bees flying around. But then look at the photograph on the right, and that shows you a rock painting from KwaZulu-Natal in the Drakensberg. And again, it shows you an individual climbing a ladder towards a bee colony and presumably harvesting the honey, and you see the bees flying around. Now, if you think about that, these two paintings are about 9,000 kilometers apart. So they represent a representation in Europe and a representation in Southern Africa. And it begins to give you an inkling about the distribution of honeybees. Because if you look at the other animals on these paintings, they are of animals that are specific to those localities. So that then brings us to the question of where is the honeybee actually distributed? And if you look at this slide, the green portion of the slide, including Europe, by the way, was the original distribution of honeybees. So it was distributed from Southern Africa to the Scandinavian countries in the north and eastwards in the Middle East. So it was a massive distribution. And if you think about it, it was distributed of a huge variety of different kinds of habitats. So it's an extremely adaptable species. Of course, starting somewhere in about 1622, some European colonists took bees from Amsterdam and transported them to New Amsterdam, which is now known as New York. And those bees then became established and spread through the whole of the North American continent. There's another story about spreading through the South American continent, but I don't have time for that. And of course, they spread to Australia and New Zealand, and they've also been introduced into China, Southeast Asia, and India, because the bee species that occur there in Southeast Asia are in fact the other 11 species. But this is an amazingly widely distributed bee. And then you need to look at modern human expansion. And here I've just shown you how Homo sapiens is thought to have expanded from Africa. And the numbers there referred to the time before the present when these expansions took place. So what you can see by comparing the two figures is that actually humans are also highly adaptable, because they're actually moving into a huge range of different habitats and moving basically all over the world. And honeybees, with the assistance of humans, have also moved all over the world. So both of these species are highly adaptable, and they, of course, are very social. I'm not going to draw any more parallels than that, because I actually want to get into the details of the honeybees. And essentially, in the case of the honeybees, you need to understand in the biological world that there are a number of levels of complexity. So at one level, you have individual cells. And those individual cells have a whole set of interesting mechanisms inside them that allow them to survive. And these cells, of course, aggregate into organisms such as ourselves. All of you are a mass of churning cells. But all of the cells in a particular organism have to be organized in such a way that they keep the organism alive. And for the most part, they do that. But in order to understand bee social behavior, what you need to do is you need to make a jump at a level above the organism. Because actually a bee colony is a superorganism. It consists of a whole set of individuals, and those individuals have no independent existence outside of being part of that colony. In the same way that your cells don't have an independent existence outside of your body. So in other words, what they've done is they've actually given up an individual existence in relation to existence in the superorganism. And actually, when you look at the reproduction of honeybee colonies, it's the colony that reproduces. The individuals do as well, of course, but we'll come to that in a moment. But it's the colony that reproduces. So when a colony swarms to create a new colony, that's the reproduction which is taking place. And if you want to measure the population sizes of honeybees, you have to measure the number of colonies to give you an idea of the population size. So the bees themselves only acquire a certain fitness by being a member of a successful colony. And this may explain the rareness of honeybee species, because this end point of the evolution of social organization is not achieved very often. And of course, for Darwin, one of the major difficulties was the honeybee, because he recognized that individuals that don't produce offspring have essentially zero fitness. And so how could that come into being? Well, he had some various ideas which were not far off the mark. But I want you to understand that what we're talking about is a superorganism as a complex adaptive system. Okay, and in order to understand that, what we need to do is we need to look at sex and its consequences. That's very important. The second thing we need to do is look at making docile workers for the good of the hive. And then we need to look at worker policing and the manipulation of communication. And then finally, the disruption of the social regulation that leads to social parasitism. So I'll go from bees in general to Cape bees specifically. Okay, let's look at understanding sex in bees, and you need to know who the players in the game are. Well, as always, it's males and females. But in the case of the male drones, they, as you see in that animation, are highly attracted to females in the air, and then they mate with the reproductive individuals, and that is essentially all they do. Because actually in honeybee males, mating is lethal. So you either successfully mate and die, or you don't mate and you die anyway. But anyway, that's what the males do. The bulk of the colony is made up of females. Males are there for certain periods of time, and they're not there for other periods of time. But the females are the key. And you have reproductive females, which you see there, which produce the offspring and mate with the males. And then you have a whole host of workers in the thousands that actually support that reproductive individual and support the colony. And you can see by looking at them that they have two quite divergent morphologies. They are completely different in size and shape and the way in which they behave. And that's a consequence of sociality. So let's look at the reproductives first of all. And you'll see that I'm not talking about queens. These are reproductives. Queens is an anthropomorphic concept. We can come on to that. But the reproductive individual in the colony is actually an egg-laying machine. She's multiply mated by those drones which you saw flying around there. So she mates between 12 and 77 of them. And what she does is she stores a sample of that sperm from those individuals and then uses that for the rest of her life to fertilize eggs. And she's got a lot of fertilizing to do because she lays about 2,000 eggs a day, depending on the season. So that's why she becomes a specialized egg-laying machine. The other interesting thing is that she has control over the sex of her offspring. Because in honeybees, if the egg is fertilized, you're a female. And if the egg is not fertilized, you're a male. So the queen has control immediately over the sex of her offspring. And what that allows her to do is it allows her to bias the sex ratio dramatically in favor of females, because she has 2,000 to 60,000 of these female offspring making up her colony. So that's a key element of the biology of honeybees that makes it possible. And then the other very important thing is that she's a generator of chemical signals that regulate the activities of the workers. And I'll come on to explaining that in a moment. Now, I also wanted to just show you this diagram, which is a drawing made by Swammerdam in 1675. And this was a eureka moment for understanding honeybee behavior, because what he's drawing here is the ovaries of the queen. And you can see the two ovaries there with very many filaments within them where the eggs are produced. And then the eggs go down those ducts. I don't know if you can see yeah, here you can see, oops, let's go back. So the eggs go down these ducts and they pass this small round gland here, which is called the spermatheca. That's where she stores all the sperm. So as the eggs go down the oviduct and past the spermatheca, she either releases 4 or 5 sperm and the egg gets fertilized, or she doesn't, and then the egg is unfertilized. So that's how she achieves her control. All right, let's take a look at the workers then. The workers, first of all, have ovaries which look like that. And you can see that their ovaries bear no resemblance to that of the queen. They are highly reduced. And so these individuals are not capable of any significant amount of reproduction. But what these workers are designed to do is they're designed to perform all of the other activities within the colony. So they have reduced ovaries with limited reproduction. We'll come on to that later. They feed the larvae in the colony. They produce the wax from which the comb is built. They collect pollen. They make honey from nectar. And they defend the colony. And then the important thing is they regulate the temperature of the colony. The temperature of the colony is maintained at about 34.5 degrees centigrade, irrespective of what's happening outside. And that's critical, because remember that these colonies depend on producing offspring at a very high rate. And one way of ensuring that you can maintain this continuous production of offspring is essentially by having an incubator, and that's what they have. And then, of course, they have a short lifespan. So they pay for all of this good stuff by not living for very long. They live for about 3 or 4 weeks, and that's partly why the queen needs to turn over individuals so dramatically. So workers work hard and die young. That's essentially what happens in a honeybee colony. Now, the interesting thing is, how are their activities organized? Now, old ideas were that there was a queen and the queen regulated anything, everything, by analogy with royalty in human societies, but actually that's not what's happening here. Their activities, which I've listed there, are partitioned by age. So at a certain age they do certain things, and as they age they move on to other things. And their activities are also determined by an interaction among themselves. So, for instance, a bee will forage a dance by another bee, and that following that dance will tell it to go out and forage at a particular place for nectar or for pollen. So their activities are regulated by age and by interactions amongst the individuals, not by some overarching control. And essentially the bees are multitasking and going through a whole series of activities. And what that enables them to do is it enables them to maintain these very large colonies. But the key is having control over reproduction. And so that brings us to the question of how do you make docile workers? Well, in the case of the honeybees, they've solved the problem by the queens producing a chemical pheromone. I won't go into the details, but I've shown you the mandibular gland pheromone there, which is the key component. And what that key component does is it makes docile workers, as you see on the right-hand side. And those docile workers do everything I told you about previously, going through all their behavioral activities. Now, in any society, whether it's a human society or a bee society, sometimes people don't get the message. And occasionally there are bees that don't get the chemical message from the queen, and they become reproductive workers, which are indicated to you there that be outside the circle. And they become rebels. Now, there are a variety of reasons why that happens. But the interesting thing is that those individuals can lay eggs, and the eggs can give rise to offspring, but only males, because they're not mated. And so you do have these rebel workers, and the problem with the rebel workers is that because they're reproducing, they're doing no work. Well, I mean, they're producing eggs, but they're actually not doing all the work that everybody else is required to do. So this is a highly undesirable situation. So what do the workers do to police these rebel individuals? Well, the main level of police, they harass them, of course, but the main level of policing is that they eat the eggs, so that the eggs that these individuals produce are not competing with the queen's production. So that's the way in which the honeybees make these docile workers and keep them in that condition, because that chemical signal, as I'll show you in a moment, is maintained by these social pheromones. Okay, so let me just quickly show you how these social pheromones operate. On the left-hand side, the social pheromone has the effect of regulating the bee's behavior. So in the bottom diagram there, when the signal is present, the workers are prevented from reproduction and they do all their other activities. When the signal disappears for whatever reason, let's say the queen gets old or she disappears or she gets sick, when the signal disappears, the workers respond by building what are called queen cells. And you can see you can see these large queen cells here, which are built on the outside of the comb to rear new queens. So the workers recognize that the social pheromone's gone and they start rearing new queens. The other thing that happens, of course, is that these social pheromones regulate aspects of behavior. So you can see that the workers form a court around the queen, and that's important for the distribution of the social pheromone, because all of those workers around her will now move through the colony, taking the signal with them. Because you can imagine if you're a queen sitting somewhere and there's 60,000 other individuals that you have to get the message to, you need a number of messengers. And these are the messengers that take the message around the colony. The other thing is I told you about colonies reproducing, and they reproduce by swarming. So you can see that this picture here is of a swarm of honeybees there next to the arrow. And they are kept together by the social pheromone until they decide where they're going to go. And then finally, of course, as I showed you in the earlier slide, this pheromone is used to attract the males for mating. So it's a very coherent system, and the colonies work very well. And you might well ask, in circumstances like this, well, what could go wrong? Well, that's the interesting question. What could go wrong? And that brings us to the Cape honeybee. And the person who first documented this disruptor, which arises from Cape honeybees, and he published his work in 1912. His name was George Onions. He was a beekeeper in Cape Town. He was also an electrician. But what Onions did was to actually do some very careful observations of his colonies. And what he did was he studied some queenless colonies of Cape honeybees, and what he observed was that the laying workers in those colonies produced additional workers, not males. Now that was absolutely startling, because everybody knew, everybody in the scientific world knew, outside of South Africa, of course, that laying workers in honeybees can only produce males. But here was this beekeeper in Cape Town saying, actually, that's not the case. These workers can produce females. And his claims were regarded as being incredible at the time, and for quite a few years afterwards, actually. Because I want to quote a small thing that was written by the South African chief entomologist in 1954. And he wrote, "It seemed incredible that South Africa should have a strain of honeybees that differed so radically from all other closely studied races of the honeybee." That was the basis of the incredulity. Okay. And of course, Onions himself was not a scientist, although his experiments were carefully done and he documented them and published them. Okay. Now, the confirmation of this production of females really had to wait until 1961. So there's a period of about close on 50 years between the discovery and this. And this resulted from the work of Dr. Anderson, who worked here in Stellenbosch, and went to Cornell University to do his PhD. And what he did in that study was to confirm the presence of laying workers in colonies, even in the presence of a queen. Now, that's another interesting thing we can come to later. But he also found that queen removal resulted in workers fighting amongst themselves and killing quite a large number of workers. It's generally fitness reducing as far as the colony is concerned. And he also found that some of these laying workers became dominant and could regulate their fellow workers. He did some careful dissections and showed that the laying workers had activated ovaries. They were producing eggs. And their spermatheca, that little thing that holds sperm, was empty. So they were producing female offspring. Now, at this stage, the scientific community, of course, began to believe that there may be something in this story, because after all, he got his PhD from Cornell. And so the interest in the actual discovery began to pick up. And interestingly enough, the understanding the mechanism by which it takes place in the laying workers themselves was done by an Indian scientist by the name of Savriti Verma, and she did her PhD in Frankfurt. In Frankfurt, they were particularly interested in Cape bees for a variety of reasons, but she was able to show that the division of the cells in the ovaries of the Cape worker bees was unusual, in that rather than just producing 4 oocytes like that were haploid and were producing males, what happened was that these 2 central ones here fused to give back the diploid chromosome number. So here we had, for the first time, a demonstration of the actual mechanism by which these female offspring are produced. Now, of course, one of the things that intrigued me was the question of why it is that these individuals that are producing females should be attractive to their nestmates. Why are their nestmates attracted to them? So, together with a colleague of mine in the Netherlands, Hajo Veldhuis, we began to explore pheromonal mimicry. In other words, the way in which these workers that produce females are able to mimic the signal of the queen. And so what we were able to do was, don't worry about the details of this biosynthetic pathway, except that there are 2 arms to it, one which is in green here, which happens in workers, and one which is in purple here, which happens in queens. So queens produce this social compound, which regulates the colony. Workers produce this compound, which becomes part of royal jelly, and that's another story. But what we were able to do is we were able to show that these Cape-laying workers were actually producing this material. So they were able to do things that normally only queens can do. Okay, so if we put this all together about the Cape honeybee laying worker, what we see in the middle diagram is that they have fairly enlarged ovaries. And if you look at that arrow there, you'll see there's an egg in the ovaries. So they are actually laying eggs. They produce this pheromone here, which is the social pheromone of the queen, and they produce it in this gland here in the head, which is called the mandibular gland. And then if you look at this diagram here, you'll see that these individuals here, which are acting as laying workers, actually have other individuals surrounding them. And so what they're doing is they're attracting those other individuals by the social pheromone that they're producing. They also produce multiple eggs in cells, as you see up there. So it's relatively easy to detect them in the early stages. Okay, so that's the sort of brief story about why the Cape honeybees are so important biologically. The next question is, where did this lead to? Well, it led to what became known as the Capensis calamity. And this was an experiment that was done by beekeepers in 1991. I mean, it wasn't an experiment, they just moved hives, but it turned out to be an experiment. So if you look at this drawing of this map of South Africa, the red portions are the Capensis bee, that's where it's distributed. The yellow portions are where the other subspecies of honeybees occurs in South Africa, and the brown portion is where they kind of integrate. But what the beekeepers locally did was they transported colonies from down here up into the northern parts of South Africa. Now, the interesting thing about this was the result of these translocations was the establishment of a social parasite. And the social parasite causes significant colony losses in that region up there. And the study of the interaction of these social parasites and how they were produced and the way in which they work has largely been driven by Mike Alsop, who works in the ARC here in Stellenbosch and who is here this afternoon. And so Mike Alsop, with collaborators from the University of Sydney and from the University of Halle, has managed to begin to unravel what this very interesting set of interactions is about and what it tells us about the interactions amongst individuals in a colony. But I need to just give you one other piece of information which is really peculiar. And that is that when Per Kruger, who is Danish, was working in Pretoria, he looked at these social parasites in these colonies in the northern part of South Africa, and he found that they were all a single lineage. They were a single lineage of individuals, and they were distributed all there. Now, if you think about that for a moment, this is peculiar. Because if you there were roughly about 400 hives that were translocated. If those 400 hives each had a queen, and each of the queen, let's say, was mated with 35 males, then the potential number of lineages that could have become social parasites is very large. And what he was saying was actually one of them hit the jackpot. And the question is whether that is the case and is still the case some 30 years later. But there is something peculiar going on here. So in other words, if you look at this map, the capensis are in the blue, the intergrade is in light blue, and then you have the other subspecies in the north. Then you have a whole lot of apiaries that are infected by the social parasite. And the social parasite, of course, is leading to colony losses, which beekeepers, of course, never like very much. And so what I decided to do with one of my students was to do an analysis of pheromonal regulation in these social parasites. And what my PhD student was able to do is that she was able to show, well, what she was doing was looking at gene regulation through the synthesis of these compounds, and she was able to show that the gene regulation is different between capensis and the other subpopulation of bees. So it means that in honeybees, the workers are not only genetically diverse. So when you look at a honeybee colony, let's say there are 35 different males in there, then that means there are 35 different families within that colony, and they're all interacting happily with each other. But if you take a look at these individuals, they're not only genetically diverse, they're also developmentally diverse. Because how do you go from being a normal, docile worker to one of these false queens? And that becomes quite complicated. So if we look at the diversity within these Cape colonies, we found in the bottom right-hand corner is the normal subordinate worker, the one that knows its place and gets on with its work. And one of the things that characterizes those is that it has undeveloped ovaries and it has all of its glands producing worker compounds, which you see in green. Then if you come here to the left, you have what are called laying workers. And laying workers are, in a sense, wolves in sheep's clothing, because what they have is they have activated ovaries which can lay eggs. But if you look at the signals they produce, they work alike. So their fellow workers don't recognize that here's something that could potentially lay eggs, because they can't recognize it. Then if you go up to the top right-hand corner, you have the incipient false queens, which have inactive ovaries, but have all those glandular components producing materials that are like a queen. So those individuals are recognized by their nest mates as potential queens, and what that recognition very often does is it leads to feeding of that individual, because they always feed queens. So they feed individuals that have these signals, and of course that activates the ovary. And then you have the full-blown false queen here in the top left-hand corner. They have activated ovaries. They have the chemical signals all producing queen-like chemical signals. So there you have a set of diversity within the workers that leads to a fairly complex situation within the colony. And so then the real question comes, how do you regulate those individuals that I've just shown you? What does the queen signal actually do? Well, if you look on the left-hand side here, you see workers of honeybee subspecies that produce males, the standard one that you find basically all over the world. And those individuals only produce these green compounds, these green compounds here, which you see on the right-hand side of that diagram, and they produce this compound, which is shown in light yellow, which is part of royal jelly. So that's part of being a regular old worker doing your thing. But in the Cape honeybee, what we have is we have workers that are kind of ambidextrous, if you want to call it that. They can produce the queen compounds, and they can also produce the worker compounds, which means they can move from one to the other. And what Fiona found was that the queens in these capensis colonies are actually regulating these workers at the level which is shown by that arrow there. So in other words, it stops them from producing the final product until the queen signal is removed. And once and so what you can think of these false queens as the people who are lining up in an F1 race. And the moment the light goes green, i.e. the moment that the queen is no longer giving the signal, then those individuals immediately start producing the queen substance and acting as false queens. So it's actually turned out to be biologically one of the most interesting things to explore. And I think that some of the key insights that come out of this is that Cape honeybee workers can mimic the chemical signals of reproductives, and they create a source of colony disruption and a source of individuals that lowers the fitness of the colonies. They also have these pseudoclonal lineages of workers that act as social parasites in colonies of other subspecies, and it leads to the death of the host. What it also leads to is it leads to the independent reproduction of those parasitic workers. So those parasitic workers have, in a sense, escaped from the super colony, because rather than their fitness being tied up with being a well-ordered member of this thing that's working, their fitness depends on being able to find another host colony into which they can go and start reproducing. So they, in a sense, have developed a mechanism for independent reproduction, and of course it gives you insights into how social parasitism might evolve over time. So the other key questions are how these potential anarchists are regulated, and we've shown you that the regulation is both by social interactions and by signals, the social pheromones. But the question still remains, why did this trait, which reduces colony fitness, get established in the Cape honeybees? And that's a question that still requires an answer. And so essentially, how do you have a normal, stable superorganism that maintains within it this lurking source of disruption? So I think exploring the reasons why this particular gene has been maintained in the Cape honeybee is going to be one of the most interesting things in the future. And with that, I just wanted to show you some of the people who have been working on these problems with me, and to thank you for your attention. I didn't take as long as Donald Trump.
Thank you. Yes, thank you very much. That was absolutely fascinating, and ended with this really, really wonderful open question, which is yet to be explored and discovered. Thank you, Robin. So, good afternoon, everyone. My name is Christoph Poe. I'm the Senior Program Manager, and like Edward said, I wanted to recognize those who are online as well. Thank you for joining us, several fellows among them. Those online can, of course, use the chat or the post function to make some comments, but we won't be reading your questions online. We'll take the questions from the room here, and those online, of course, afterwards, Robin can have a look at your comments and questions as well. So we have time for, Robin, do you prefer taking a question at a time, or two or three at a time?
I think it's easier to take a question at a time.
Right.
Yeah.
We'll do that. We'll do that. So let's see who would like to start us off. Yes, Jon will start right to the third row here. Please do use the microphone for the sake of the online audience.
Thank you for a fascinating talk. You started off by talking a little bit about the spread of bees and the human impact of. I was wondering, how much can bees spread just intrinsically by themselves? So what distances, do they spread?
You mean, how did they spread?
I guess humans brought them, didn't they? Or they'd spread naturally?
They spread naturally.
Okay,
Yeah. I mean, there's a debate about whether the ancestor of the honeybee emerged in Africa or whether it emerged in the Middle East. And that's an unresolved debate. But in fact, having emerged wherever it was, they diverged into lineages that went into Europe and covered Europe, and to lineages that went into Africa and covered Africa, and also got to Madagascar. So that was the original distribution from southern Africa to Scandinavia, and then off to the Middle East, probably to the edge of Russia. But where you find them in North America and South America and Australia, they've been taken there by the bees used humans to actually expand their range. Yeah.
Two further questions, John, and then Ilva. So you can go ahead.
Really, really interesting, Robin. Thank you. Do we know within the Cape honeybee how these special, potentially social parasitic workers are treated in terms of their, are they followed as well and fed as well, and are they reproducing well? I guess the question being, why aren't they killing the Cape hives, Cape colonies?
Can you just repeat it for me, John? I didn't hear very well. Sorry.
Oh, sorry. Have you or have others studied how these social parasitic workers are treated within the Cape honeybee colonies?
Oh, that's a question for Mike to answer. But I think that the Cape honeybee queens appear to be capable of regulating the workers in their colonies, but a queen's ability to regulate fluctuates through a whole lot of factors: the size of the colony, age of the queen, and so on. So exactly what happens is we're not sure. And the other interesting thing is that if there was such strong selection for a single lineage in that initial thing, then what that suggests is the virulence of individual lineages is actually quite variable. So that appears to be a particular lineage which doesn't get the message, if I can put it that way, and is able to go off independently, whereas other lineages may be easier to regulate. But that's speculation.
Great. Thank you. Ilva is next. Yeah.
Thank you. Thank you very much, Robin, for this really fantastic presentation. You started to talk about the superorganism, and I'm thinking that as part of that, we could also think of the microbes that they are carrying. And I know that bees are also carrying their own very specific microbial flora that they need.
Yeah.
I haven't followed that field at all, so I don't really know what they use the microbes for, I guess for nutrition and so on. But is there anything known about if they also use the microbes to produce some of these chemicals, like, that the microbes could provide some of the enzymes? Because I'm thinking of evolving a new pheromone system or so would take very long time in evolution, but if it actually could be brought there by some microbes, you could have very quick changes, and one could imagine that this could also be sort of a parasitic thing that could come into a colony, that some of the individuals actually could carry some microbes that then produce a pheromone that acts a little bit differently, and that this could be very, very quick change from an evolutionary time perspective. But is anything known if this can occur?
Well, the work on the honeybee microbiome is actually very recent.
Yeah, it is. I know.
And it's mostly in relation to the way in which various pesticides actually have an influence on the microbiome of the bee and therefore on their health. But the question that you ask is different from that entirely, and I don't know of any work that's been done to explore that. I mean, one of the things that's interested me is that these social pheromones, which are used by the honeybees, are entirely unique, and are unique to honeybees. Because social pheromones in other social insects, not the termites, other social insects like ants and wasps and so on, are a different class of chemicals altogether. So this is a de novo development in honeybees, and we've also sampled a whole range of other bee species to see whether you can find these compounds, and you can't. So the other bee species don't produce anything which is equivalent to this. And so that's all.
You have mapped the genes, so you know that these are produced
that's
by.
right.
Yeah, you have the genes there.
Yeah. So I mean, there might be a microbial evolutionary background to that, and nobody's explored that. Yeah.
Okay, great. More open questions. We have in the front here, yes.
Thank you. It was fascinating. And I know you kind of spotlighted the anarchist in the group. But I'm intrigued about this sort of transition moment. I mean, how long does the queen last? And kind of what happens there? Does that lead to a breakup of the colony? What are some of the dynamics?
Okay.
Yeah.
The queens lasts somewhere between 2 to 4 years. But the other thing is that when colonies reproduce, the first swarm that's produced by a colony takes the old queen with them. So, I mean the interesting thing about bees is they are unsentimental about the aged, if I can put it that way. If a colony needs to defend itself, it's the older workers that go out and defend the colony, not the younger ones, which is the reverse in humans. But to come back to the queens, the old queen goes off with the swarm to start the new colony, which is inherently risky, and she leaves the established nesting site in the possession of her daughter. And so the daughter is then a young queen which takes over the colony. So that's how it works. And in a study that we did just north of Pretoria, we discovered that about 80% of the colonies these were wild colonies in a region where we looked. There was an annual turnover of queens, which you can do genetically. I mean, there were two interesting things. One is that there was this high annual turnover of queens. So when you look at a colony like that one, which I showed you, and you come back in 3 months' time, almost all the workers are different from the ones that you photographed, for a start. If they swarmed, then the old queen is gone and her daughter is taken over. So there's a high rate of turnover in these colonies, and in some instances we found that the turnover in the colony was by a foreign queen. So the old queen had left, leaving the daughter in place, but the daughter was actually usurped by an individual who came in from the outside and took over that nesting site. So there are an interesting set of interactions which go on in there. Yeah.
Thank you. That was really fascinating. I just have a question. I didn't quite understand why you use the terminology of social parasite. And I know you said it's losses in the colony, but what is the actual social behavior that they're doing that leads to the losses? I understood the diagrams that you did, they were producing the pheromones, but what was actually causing the loss, and why the concept of the social parasite?
Okay, when the social parasite gets into a host colony, it's dependent on the host workers to rear its offspring. Usually the queen in the colony disappears. So there's nothing in the colony which is producing new host workers. So these host workers age and eventually disappear. But those host workers are necessary to produce the offspring from the social parasite. But all of her offspring, of course, want to be social parasites. They don't want to work in the colony. And so what you have is a situation in which, as the host workers start falling off and dying, then those social parasites need to find another colony in which to rear their offspring, because they themselves are not good social actors. You know?
I just want to, you said there were 12 species of honeybee, and the species that is affected with this phenomena is of the same species but a different family.
You mean in social?
The Capensis and Sodine scutellata are different families. Yeah. Well, what I'm trying to find out is, in the same species, why did this characteristic develop if it was the same species?
Yeah, I mean, this is developed within Apis mellifera, which is one species, and intraspecific social parasitism is exceptionally rare. Most other social parasites, whether they're bird social parasite or insect social parasites, are actually different species from the ones that they're parasitizing. And so this is a very unusual situation. And also it's not clear whether this form of social parasitism is going to survive evolutionarily. Because in essence, the social parasites are being perpetuated by beekeeping, because the colonies are collected in apiaries. But in colonies in the wild, it appears that the colonies are, for the most part, are far enough apart that the social parasites have difficulty in finding another colony.
But why is the social parasite not a threat to the Cape honeybee?
Why is it confined to the Cape bee?
No, why does this parasite not pose a threat to Cape honeybees, but it poses a threat to the bees moved up when it moved up to the north?
Yeah, it's John. That's a very interesting question. I think that it has to do with the way in which the Cape worker bees treat their brood, and the way in which the Scutellata bees treat their brood. So one of the things that it seems is that the Cape honeybees, if they lay an egg in a Scutellata colony, that gets fed. In other words, there's a larval pheromone that results in that individual being fed, and the likelihood of it becoming reproductive going up very significantly. Whereas that appears not to be the case within the Cape honeybee. So there's a level of regulation going on there that we don't fully understand. That's what Mike's going to solve for us.
Thanks. What is the effect on you say the other colonies are dying off? But does it benefit the Cape honeybee? The lack of colonies then elsewhere, do they spread? And secondly, is this phenomenon of social parasites leading to a colony die-off possibly related to the mystery around bee colony die-offs?
No, I think those are completely unrelated. I mean, I think that the colony die-offs which you find in California, which is related to almond pollination, is related to a whole range of factors, but it's completely different from the social parasitism that we see here. So I don't think there's any relationship at all. Yeah.
Yeah, thank you. I like the way you characterized social parasites as potential anarchists in order to make us understand that all the other bees are actually pure fascists, I suppose. Especially as you ended your talk in mentioning Trump. I think it's food for thought. But my question is about numbers. You said that there are 20,000 different species of bees in the world, and just a few of them are honeybees. But in terms of numbers of individuals, as honeybees have spread all around the world, are they more numerous than the other bees or not? And how do they cope with, let's say, pollution and global warming and etc., the differences between honeybees that are really very much related to human exploitation and wild bees?
Can you just...
comparing the number of honeybees population-wise with the other 23 other species.
Yeah.
And then the climate impacts.
Yeah. Yeah. Look, I mean, the population of honeybees at the moment has probably never been larger. than ever in the past. And that's partly due to two reasons. One is the translocation into new habitats in North and South America and Australia and so on. The other one is the massive increase in beekeeping activity. So the number of colonies of bees that are used by beekeepers has gone up dramatically in the last 40 years or so. So, and since the colonies are the population, that's a very good indication that's gone up significantly. Now, the interesting question that we don't know, because we don't know much about the wild populations, is the degree to which things like the intensification of agriculture, climate change, and land use changes are actually having an impact on that large population. So that's a very large unknown. And I mean, thinking about it, there's no reason why all of those factors shouldn't actually be fairly dramatically reducing the size of the population. But then I think is one of the really interesting areas where people need to be exploring in much greater detail, because the other question that arises is that if you need to intensify agriculture in order to support the increasing human population, then it places a huge demand on pollinators to increase that food production. And we have no idea whether there's sufficient numbers of them to actually be able to scale it up. It's not as simple as simply ploughing a land and planting the crops. You've got to actually have the bee colonies to do the job. And that's also one of the big questions, particularly in countries like the Southern African countries, which are relatively arid and in which we think the bee populations are relatively sparse. Whereas in Europe and various parts of North America, the wild populations are actually quite healthy, even though there's an IUCN red data list for the bees in Europe. Yeah. But that has to do with the intensification of agriculture for the most part. Yeah.
Thanks. We had Amrita.
Is it working? Thanks, Robin. Yeah.
Oh, there we go.
Thanks, Robin. That was fascinating. So my knowledge of bees is limited to this presentation and a fascinating story by Roald Dahl called Royal Jelly. So forgive me for this question, but I was fascinated by bees because of that story by Roald Dahl, and it was called Royal Jelly. So I want to get your help in some sense passing out fact from fiction. In that, Roald Dahl had said that queen bees rarely leave the hive voluntarily, but they are forced to leave it by the workers when the queen bee stops producing enough eggs. And the way that they do that is by refusing to feed her royal jelly. And that makes her thinner and thinner, and then she's pushed out of the hive. So that could be purely Roald Dahl's imagination. But I also do want to hear from you if there's any bit of truth in that. And secondly, what is this royal jelly? So in that story, a man keeps feeding his baby royal jelly, and royal jelly apparently has a huge market in human something. So could you tell us more?
Let me say there's a lot of hype about royal jelly, most of it with no substance. But royal jelly is actually a glandular secretion that is produced by the worker bees, and which is used to feed the young larvae. And the difference between a queen and a worker depends on basically how much of that is actually fed to them in those first few days of their life. Now there's a huge literature in which everybody went on a mad hunt for the magic queen determinator that was supposed to be in royal jelly, and which, if you got it, you would suddenly become this reproductive, and if you didn't get it, you were stuck with being a worker. Well, after many years of hunting, that's not been found. And so what it really boils down to is a question of differential feeding. It's the proportion of royal jelly relative to other components that go into the diet, which the workers actually feed to the larvae. There are other things about royal jelly which are chemically very interesting, because it's very important in rearing queens. Because as I showed you in the picture, the queen cells hang downwards. They're not horizontal like the normal cells are, so they hang downwards. So the queen has to lay an egg at the top of this thing which is facing downwards. And so how come the egg doesn't fall out? Well, actually the egg doesn't fall out because actually when they make the royal jelly, it makes a kind of a glue. So part of those chemicals there are a glue which allows the egg to be stuck to the top of the cell, and that's a really interesting. That's why the workers have to produce that other compound, because that other compound is critical for making the glue. Otherwise, you don't have queens.
Okay. We have to end it there. I don't think we have royal jelly for you, but there is some wonderful other things waiting for us. Before we do that, I just wanted to acknowledge and thank everyone who came to the lecture, all the questions, everyone who joined online. And Robin, wonderful. Thank you for this really, really insightful lecture and presentation. Thank you.